Hello, my name is Sanjay Saraf. I'm a student in E25. This is my bonus project on watching the flu. You'll have to excuse me, I'm a little bit under the weather, so I guess this project is appropriate. Here is a person who is experiencing the flu. We as humans usually experience the flu year to year. It's a seasonal flu, generally. When it isn't a seasonal flu, the swine flu and the bird flu in 2009, 2005, respectively, the 1918 Spanish flu, these are usually called pandemics because they have a higher death rate and they spread through the population much more quickly and much more devastatingly. So that's what this project is about. So here's a general overview of the viral particle. We have two main surface proteins. That's going to be the focus of this particular project. Hemagglutinin, which allows the influenza virus to enter the cell. These are the yellow spikes. And neuraminidase, which are these green and red mushroom proteins that allow the viral particle to leave the cell once it's been infected. As Cliff mentioned, the general overview for a viral particle has this lipid envelope and a capsid which contains the nucleic acid in the middle. So this is a close-up view of hemagglutinin. I can talk a little bit more about the science of it, but essentially this thing works by inverting itself. So if you picture this sort of shaped like a sock, and it flips inside out, and that's what allows the virus to enter the cell. Okay, so here's some numbers on the three most recent pandemics, or the three most recent really devastating pandemics. We have the 1918 Spanish flu, the 1957 Hong Kong flu, the 1968 Asian flu. They generally killed lots of people. The way these flus worked is that they used reservoirs in either pigs or birds or horses to recombine with the human influenza virus. And it only takes a few influenza viral particles to really recombine in a nasty way for it to retain the ability to transfer from human to human, but also have sufficiently different genetics so that it's not recognizable by the human immune system. And so that's how sort of these pandemics come about. So engineers have sort of come up with different tools to try to fix the problem of pandemics. One of those is called Flu Tracker. Essentially what Flu Tracker does is it looks at news stories and reports and tries to map the presence of flu in many different places. So let's see if I can bring it up and show you what it does. Okay, so here's Flu Tracker. Essentially we have what's a Google map, and it has the number of flu cases and it sorts them by location. Now one issue with this is that it's not very clear what sort of flu they are observing. And what you often see is that they have all of the reported flus in their data, where we're really only concerned with the pandemic flus, right? So a lot of this is the seasonal flu that's been mixed in. And so as you zoom in, you'd like to say, well, okay, maybe I could say something about the presence of flu in California, but I really can't, because we have far more data than we have far more seasonal flu than we have pandemic bird flu or swine flu that we think might be here, right? And so the data gets, it's just too much data, and so we want to fix this problem as engineers. So how do we fix it? Well, conveniently, we actually know the genetics of the flu viruses that are being isolated. Flu Tracker doesn't use it, but those genetics are available to us, and we can use those genetics to determine whether it's a swine flu or a seasonal flu. And so we have here this organization called NCBI, which provides this database called GenBank, which contains the genetics for isolated viral particles in different places. Okay, so here's an example, and this gets to what Cliff was discussing about point mutations and frame shift mutations. And because influenza is a virus, for a few different reasons, it actually mutates much more quickly than, for instance, a native mammalian cell. And because it mutates much more quickly, it has the ability to evade immune systems much more frequently. And you see here sort of different flu viruses having different point mutations in the things that aren't colored green. Okay, so how do you actually determine the difference between two sequences quantitatively? And the way that we do that is we use something called a Levenshtein distance matrix. The way we functionally do it is that we're looking to see how many differences there are between sitting and kitten, for instance. And by observation, we could say there's 3 differences. You switch S and K, you switch I and E, and you take a G and add it on. Right? So that'll make sitting from kitten, and vice versa. The way we do it algorithmically is that we sort of list these guys and then dynamically increase these numbers depending on the correlation between these letters. And so we're dynamically making a matrix, and at the end of the matrix we can read off what the number of differences between these two words are. Alright, so another tool that's useful for us as engineers is something called Google Maps API. So you're familiar with Google Maps probably. The Google Maps API gives us an ability to map things in code that we wouldn't normally be able to do. And so it's very useful for constructing data and then displaying it. Okay, so here are my references. So let's show a quick demo of where I got the data from and then what I did with it. Alright, here we go. So here's an example of a GenBank entry for a particular virus. So this would be the title of the entry. And we were using segment 4, which is hemagglutinin, the protein that's allowing the virus to enter the cell. And this is the protein that changes the most, generally speaking. So we can look and it tells us a little bit about the virus, which we know is generally true for influenza viruses, but it also tells us some stuff that's specific for this particular isolate. So in this case, we know that this strain was isolated in California, specifically in Alameda. And we know that it was collected on April 27th, and that the person who had the flu was a male who was 28 years old. So we actually know quite a bit about this particular flu virus, and we actually know its sequence. So what we can see is that we'd like to be able to say, okay, we know this is a swine flu because we have the genetics, and we know where it's located, and we know when we isolated it. And so now we can use the Google Maps API to plot the data. So here's a fusion table. This essentially allows us to put the sequence data into a place where we can query it remotely from a site. And here it is. I'm going to clear this so we can show what exactly we're doing. So this is just a general Google map, as you're familiar, right? For now, I've only put about 7 sequences into California. Putting more in is sort of a trivial job of just copying and pasting from Biology Workbench into California swine flu data. And we can click on these particular red dots and look at a swine flu that was, in this case, a 42-year-old male swine flu isolated on April 26th, 2009. We can copy this sequence and we can compare it to another swine flu that was isolated somewhere else on a different day and see how different they are. 4 differences. Right, and so that tells us a little bit about how the swine flu is moving. And that would be useful for us to know as engineers and as people who are sick. So, hope you like the project. Definitely you can come visit it at sanjayvsaraf.com/flumap. Thanks.